Hi, I'm Tim Hunter, and thank you for coming to my presentation on the dynamics of the microcirculatory blood flow response. Sepsis, or septic shock, is a full-body inflammatory response to infection. It is a very common disease, and the few existing treatments, such as fluid resuscitation, are very controversial with high failure rates. It's been shown that in sepsis, the microcirculatory blood flow regulation is dysfunctioning. So why is it not treated at the microcirculatory level? We don't know how. Fundamental research on the function of microcirculation and how it can be disrupted in sepsis is being conducted in the Goldman lab at Western University. In particular, my project is focused on studying the dynamics of healthy microvascular responses with the goal of comparing these findings to research on disease cases. Study of the microcirculation is difficult due to the scale, which is on the order of micrometers. The structure and function can be studied using microscopy. However, they are intrinsically linked to oxygen concentration on the tissue, which can't be measured on this scale. Because of this, computational models are often used to supplement and inform in vivo data. We use a mouse preparation, shown in Figure 1, to observe the skeletal muscle microcirculation while the tissue was exposed to an oxygen challenge. Similar imaging setup and corresponding microcirculatory networks can be seen in the submission 2044 by Barry Jansen if you're interested. The oxygen challenge was a controlled change in the PO2 that the surface of the tissue was exposed to, as shown in Figure 2. The decrease in PO2 causes a flux of oxygen out of the tissue, which elicits a blood flow response in the microcirculation of the tissue. Using combined hemodynamic and oxygen diffusion models and a geometric approximation of the tissue, I am simulating the precise conditions of the experiments performed on healthy mouse tissues to determine the impact of the oxygen challenge on the PO2 within the tissue. If you see submission 508, my colleague Baron has been working on a very elegant solution for calculating PO2 distributions within a tissue using minimal computing power. At the top left is a time plot of the PO2 in our simulated tissue block over the 50 seconds following the oxygen challenge. As you can see, there's a dip in the oxygen pressure following the start of the challenge, lasting about 15 seconds until it is restored to a steady state, with the minimum PO2 marked by A occurring 6 seconds after the start of the challenge. After the recovery, the mean oxygen pressure was only 2 millimeters mercury lower than before the challenge, with the imposed surface pressure dropping by 16 millimeters mercury. The PO2 distribution before and after the challenge start is illustrated at the top right figure. The decrease was so small due to a 68% increase in oxygen delivery by the capillaries. In the bottom figure, the mean oxygen pressure at the depth of the observed capillaries is compared to the blood velocity and hematocrit. The plots on the left show the properties plotted over the time of the step down in oxygen pressure, while the plots on the right show the properties plotted during the step up. On the left, a fairly quick blood flow response can be seen after the decrease in oxygen pressure, with velocity increasing after 2 seconds and hematocrit after 5. On the right, the response is much slower and more gradual, which indicates that the response is only stimulated by low oxygen pressure rather than high. The key findings from this project were that the healthy blood flow response can recover the tissue to healthy PO2 under imposed oxygen stress. There is a slight delay of 2 to 6 seconds from when the PO2 drops to when the blood flow response is activated, and also that the difference in response rate to higher and lower oxygen indicated that the trigger is low oxygen and not high oxygen. I believe that the delay before the blood flow response is activated may be the key to understanding how microcirculatory blood flow is dysregulated in sepsis. With many or all tissues requiring increased blood flow, the regulation mechanism may be overloaded, and an exacerbated delay could be the cause for highly oscillatory and irregular blood flow in the microcirculation. Thank you for listening.